Hello everyone. Imagine the DNA as a long instruction book for the cell, but the cell does not read the whole book at once. It focuses small sections called transcription units. What is a transcription unit? A transcription unit is a stretch of DNA that contains all the information needed to make a single RNA molecule. It is like a mini instruction manual inside the big DNA book. The parts of a transcription unit. First, promoter. This is the starting point, a special DNA sequence where the RNA polymerase binds to begin transcription. Think of it as the green light that tells the enzyme, start copying here. Second, coding region, the structural genes. This is the main part of the transcription unit, the sequence that will be copied into RNA. It contains the instructions for making a protein or functional RNA. Third, terminator. At the end of the transcription unit is the terminator sequence, a stop signal that tells RNA polymerase when to stop copying and release the new RNA molecule. So RNA polymerase attached at the promoter move along the coding region, making RNA and stop at the terminator. This whole journey from promoter to terminator is the transcription unit in action. Why is this important? By organizing genes into transcription unit, cells can control exactly when and how much RNA is made. Now the three processes of transcription: initiation, elongation and termination. Let's dive deeper into how transcription works. Initiation. First, RNA polymerase, the enzyme that copies DNA into RNA, scan the DNA until it finds a promoter, a special start signal on the DNA strand. Think of this as a start here sign on a racetrack. In eukaryotes, RNA polymerase can't do this alone. It needs a whole team of helper proteins called transcription factors to recognize the promoter and guide it in. Once RNA polymerase is in place, it unwinds a small section of DNA helix to expose the template strand, ready for copying. Second is elongation, where RNA polymerase acts like a copy machine, moving along the DNA template strand and building a new RNA molecule, one nucleotide at a time. It reads the DNA code and strings together the matching RNA base. A pairs with U instead of a T because it is RNA, C with G and so on. The RNA strands grow longer like a ribbon being pulled from a spool. Third process is termination. Finally, termination happens when RNA polymerase reaches a special sequence on the DNA that tells it, stop copying. At this point, the enzyme releases the newly made RNA and detached from the DNA. The RNA molecule then goes on to play its role, whether to make proteins or perform other tasks in the cell. In detail, the transcription process occurs by number first, row dependent, and number second, row independent termination. Row dependent termination. In this method, a special helper protein RHO row comes into play. Row attached to the newly formed RNA and start chasing after the RNA polymerase. When the polymerase pause at a specific DNA sequence, think of it like a checkpoint. Row catch up and uses its molecular motor to pull the RNA away from the DNA, forcing the polymerase to stop and release the RNA. It's like a referee blowing the whistle to end the game. Next is row independent termination. The other way to stop is built right into the RNA sequence itself. Here, RNA folds back on itself, forming a tight hairpin loop followed by a string of uracil bases. This structure acts like a signal flag that causes the RNA polymerase to lose its grip and fall off the DNA. No extra helper needed. It is an elegant, self-contained stop sign. The RNA is made from the antisense strand of DNA. The RNA looks almost exactly like the sense strand, but with one difference. RNA uses a letter called U instead of T. To sum up, transcription is a finely tuned process where the cell makes RNA copies from DNA to produce proteins. In prokaryotes, genes are mostly on and controlled by repressors. In eukaryotes, it's more complex, requiring many helpers and control cells to turn genes on or off at the right time. There are 3 RNA polymerases. RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 2 transcribes protein coding genes into messenger RNA or mRNA. RNA polymerase 3 transcribes transfer RNA and some small RNAs. Now the processing of RNA transcript. Unlike in bacteria, eukaryotic RNA transcribes must be processed before leaving the nucleus. 3 processes, 5' cap, splicing and poly-A tail are there. These modifications protect the RNA from degradation, help it exit the nucleus and prepare it for translation in the cytoplasm. Now the first process, capping. As the RNA chain grows, the very first modification is the addition of a cap at the 5' end. This is not just any cap. It is a 7-methyl guanosine structure linked in a unique way to the RNA, forming a protective seal. This cap survives multiple vital functions. It protects the RNA from degradation, helps ribosomes recognize the message for protein synthesis, and even play a role in transporting the mRNA out of the nucleus. Second, polyadenylation. The tail that means stability, tagged with a tail made of around 200 adenine nucleotides called the poly-A tail. This tail is added after the RNA polymerase encounters a specific sequence known as polyadenylation signal. A short AAU AAA code that flags where cleavage and tail addition should happen. Why the tail? This poly-A tail act like a stabilizer, protecting the RNA in the cytoplasm and assisting in its export from the nucleus. Next is splicing. Now the heart of RNA processing. To create a mature RNA, the cell must precisely cut out the introns and join the exons seamlessly. This is no small feat because any mistake could result in faulty proteins. Surprisingly, the exact sequence marking where introns start and end are minimal, usually beginning with GU and ending with AG, but the cell still knows exactly where to cut. A molecular machine made up of small nuclear ribonucleoproteins or SNRPs. These SNRPs recognize the intron boundaries, being the exon together, and catalyze two key reactions that move out the intron in a lariat shape and join the exon into a continuous coding sequence. Exon and intron. A fundamental difference between eukaryotes and prokaryotes is the presence of intron, known coding sequence within eukaryotic genes that interrupt the coding regions or exons. Back in 1970s, the scientists Philip Sharp and Richard Roberts made a groundbreaking discovery. When mRNA pairs with its DNA template, loops appear where introns are skipped, revealing that these interruptions do not appear in the final message. Imagine the gene of a complex puzzle. The exons are the piece that matter for building proteins, while introns are the extra segment that must be removed.